Okay, our next presenter is Dr. Sandip Patel. He is the Associate Professor at the University of California at San Diego, co-leader for Experimental Therapeutics, Deputy Director at the San Diego Center for Precision Immunotherapy, and Assistant Director of the Clinical Trials Office. I'm not sure how you even have time to be here, but thank you. Please welcome Dr. Patel.
Thank you, and thank you for this opportunity to present to you this morning. It's always a treat to come here to SWOG and see how fast we're growing and all the great science and in all the ways we're helping patients. I'm here to present DART from the Early Therapeutics and Rare Cancers Committee on behalf of my co-investigators, Dr. Chai and Dr. Kurzrock, as well as the really large team that really has helped put this together. And as I stare into the lights, I actually can't see my own SCs if they're there in the back, but I may try to call them up because they deserve all the credit at the end. And so more broadly, why do we do this study? And so rare cancer studies have been very difficult to conduct. The mantra has been they don't accrue. And what happens is it's kind of a balkanization. It's a bunch of rare cancer types that it's very hard to do these studies individually. But if you actually take all rare cancers and look at them holistically, it's almost a quarter of tumors. And so it's a major unmet need. And additionally, rare tumors tend to be more frequent in younger patients younger than the age of 40. In fact, quote-unquote rare tumors are more frequent in this population. And so not only an unmet need, but unmet need in a younger patient population. One of the reasons these studies are so difficult to do logistically is because of the myriad of different histologies. And in fact, as we conduct this study, there are tumor types that I've never heard of that I had to look up and try to triage, and that's true for many of us. And so the idea that these kinds of basket studies can be done was one of the main goals in S1609 or DART. But this shows the complexity in managing all the different cohorts. And when you guys are asking us questions about which cohort the patients go into, this kind of demonstrates some of the complexity that we encounter. And so as a group, rare cancers are almost a quarter of all new cases. They're individually rare, but collectively a quarter of patients. Underrepresented in clinical trials, given the mantra that these trials won't accrue. And in fact, I think one of the most important things we've discovered with DART is that if these trials are structured appropriately, there are no issues with accrual, except potentially over-accrual. And then you have rare cancers that disproportionately affect younger patients with limited trial options. And there are a lot of regulatory hurdles for these studies as well in terms of industry. And so the cooperative group mechanism, and I think SWOG in particular, has had the foresight to focus on these kinds of studies within a committee. And I think that's led to the success of DART, which really goes to the SCs and many of you guys in the room who've contributed to this study. And so one of the premises of what type of therapy makes sense in such a study, in a basket study looking at immunotherapy. And we know that anti-PD-1 approaches work in rare cancer types. Merkel cell, anal cancers are common. The paper I'm showing from anal cancers actually through SWOG, Kathy Eng's group. And so we also know that combinatorial immune checkpoint blockade tends to have a higher response rate than single-agent immunotherapy. We've seen this in melanoma, in lung cancer potentially. Next year we'll have an approval, and in renal cell carcinoma, in which combinatorial therapy may be preferred. And so for a small study, in which we're looking at a variety of different cohorts. The overall study may be large, but each cohort individually is small, which I'll show. The idea that using a combination approach to best find a signal if there's going to be a response was the reason we chose dual anti-CTLA4 anti-PD-1 blockade, nivolumab, ipilimumab, as our backbone regimen. And the primary study objective was to evaluate the overall response rate in patients with advanced rare cancers across a variety of different histologies, as well as secondary endpoints related to toxicities, overall survival, so on. And so the way we came up with our list of histologies was through the Rare Cancer Consortium of the EU, which defines these as a consensus of less than 6 per 100,000, incidence of less than 6 per 100,000 persons per year. And then we had to exclude studies for which there are ongoing NCI-CTEP studies at the time of the protocol being developed. anal cancers, lymphomas, Merkel cell, pleural mesothelioma, sarcomas, thymic carcinoma. And those of you that have been a part of this study for a long while remember the complexities for the integration with NCI match. That has, since Amendment 4, I believe, not been an issue for a while, as this study is now freestanding. And so any kind of relics in your database or your notes about patients having to go in NCI match first, that was a much older version of the study. And so these are all the different rare cancer types. Many of these cohorts have accrued completely, and so I'll show you the best way and even what I use to keep track of what cohorts are open or not when we're trying to answer your questions. But more broadly, what kind of patients can go on the study? We try to be a big tent study in terms of the rare cancers that are enrolled, but also in terms of the types of the patients that can be enrolled. And so, for example, for brain metastasis, we do allow these patients as long as they've been off steroids for 2 weeks. They do have to have measurable disease. Patients could have had prior PD-1 or CTLA-4 before, as long as they've had over a 1 month washout, and as long as they didn't have a high-grade IRAE on study. And we do allow patients with HIV and viral hepatitis as long as they're otherwise controlled virologically. And so what's the overall study schema? There are lots of different ways to give nivo-ipi, and we have a different way, though with the potential approval in lung cancer, this may start to feel more familiar next year. So as mentioned, this is a basket study. All the arms get the same regimen. We do the low-dose ipilimumab, 1 milligram per kilogram, IV every 6 weeks, continuous. So this is different than what you're used to in melanoma, right, which is a higher dose, 3 mg per kg, or in renal cell, which is lower dose, where you give 4 doses, right, and then you just do nivolumab by itself. Here, the ipilimumab is continuous every 6 weeks, with nivolumab fixed dose every 2 weeks. In patients who have an IRAE, that the study, the local PI, as well as the patient view as something that they want to continue on therapy, there is the option for most of these IRAEs to continue on nivolumab monotherapy after management of their IRAE with steroids. The treatment cycle length is 6 weeks, and so that's kind of where the CAT scans line up at every 12 weeks. And basically, patients can continue until progressive disease, unacceptable toxicity, or withdrawal of consent. Getting into the detailed statistical considerations, and this is a study in which not only the study coordinators have been very much influential in making sure this has been successful, but the statisticians, because of the complexity of the study. It's a Simon two-stage design. So what that means, and this is why you often get an answer when you email us about cohorts opening that we don't know, is because it's a two-stage design, right? And so basically patients go on study. The first stage has 6 potential patients per histologic subtype, and we have to have a response in that first 6 in order to get 10 additional slots as part of the second stage. And so we're not trying to be coy when we respond that we don't know when or if slots will be available, but this is the statistical reason why it's a Simon two-stage design, and so we're not even sure if those slots will necessarily be available if there are no responses. And then out of 16 patients, we're looking for 2 or more responses, and we're looking by overall response rate. And so I had to lock these slides at the 1st of September. So DAR was activated January 13th. The first patient was treated at the end of that month of 2017. And then as of September 1st, we had 809 sites approved, 525 patients enrolled, additionally 37 cohorts, and now it'll be up to 53. And this, if there's one slide to really take away, or one thing to write down, it's this. It's the URL for the SWOG accrual. It shows in real time which cohorts are open, which ones are closed, which ones are permanently closed. And this is what we go off of. It's so up to date, and the statisticians and the programmers did such a great job with this that this is what we use. And so you have access to this as well. And so this tells you what cohorts are open on the most recent amendment, and so really can't congratulate the team enough for putting this together. When we first started this study, and those of you that have been with us the whole way, especially on the SWOG side, there were a lot of complexities with the number of arms, and this really made it much easier to conduct the study and keep track of everything in real time. And so kudos to the team for putting this together. And so I would recommend this is something, if you have this study open, to look at this website. I actually have it saved it as a bookmark on my phone because we get questions that often. And so very much these are some of the revisions that were in Amendment 5, which is actually currently under review, that will be reviewed at CTEP. The reason I'm slowing down talking here is actually was reviewing this revision last night for submission, and most of these are accurate. There is a choriocarcinoma cohort as well, which because of the way the protocol was worded, we use beta-HCG as a pregnancy test, and so by definition with choriocarcinoma that would be an exclusion. And so we've actually invented new immune response criteria with our colleagues at the NRG for that cohort, and so you may want to pay attention to that, but that's actually something that we'll be submitting to the NCI very soon. And then there are a variety of logistical questions. I think if there's a second thing I could recommend highlighting from these slides is in terms of logistics. If you have any questions, feel free to email the SC email, the S69SC@swog.org. You can, of course, email myself or any other SCs directly, but if we're out of town, we're off the grid, you need a response, someone will be covering this email. And so I just want to mention that we're happy to take the direct emails, but kind of like a pager, sometimes you want to sign it out in case you're flying or something, and so this is the email to go for. Otherwise, the other questions I can probably answer off the bat, and I'm happy to take them towards the end as well, is with the NOC cohort is being restructured based on NCI guidance. NOC, not otherwise classified, is not meant to be an overflow cohort. So if a cohort's already accrued, it's not meant to be an over-accrual cohort. And then one question we used to get a lot of, but I think sites that have dosed have not had this issue, is nivolumab is administered prior to ipilimumab, which is the way it's done in the standard of care setting, and that we basically use the baseline weight unless there's a greater than 10% change. This is where you can find all the other sites participating in DART at the clinicaltrials.gov site. We have over 800 sites, and so that's the clinical aspect of things. I do want to focus a bit on the translational medicine part, and this is something that Dr. Young Chai has been leading, and so deserves the credit for this. And so we've been very blessed to have a lot of correlate biomarkers, and Children's National is where these specimens are being housed, and we know you guys are working very hard to get us tissue and blood. But why? And so this really focuses on all the different assays we're trying to do. I think the overarching point, this is the nitty gritty on the slide, but the overarching point is many of these rare cancers, there's not a lot known, or there may be zero publications about in terms of their genomic characterization, transcriptomics, the immune infiltrate, and so the goal of creating a TCGA for rare cancers out of this study is really central, and so that's why the tissue and the blood are so important, because we'll be looking at both tissue-based as well as peripheral blood biomarkers. I think one of the things that has come along during the conduct of this study has been the collaboration with CMAX. And so with us, it's MD Anderson within SWOG, and Ignacio Estuba is leading this collaboration. But the idea that the translational science is really strengthened by this NCI-funded mechanism that is really investigating immunotherapy. And so DART is one of the initial studies that'll be utilizing this mechanism. And so just like when this study first started up and we had to learn kind of on our feet about how to allocate across the different cohorts, I think in terms of CMAX, so hopefully we'll be able to have some useful insights to share with everyone in terms of how best to do the correlates, which are just so important to help drive the discoveries, to help these patients, because there's just not a lot known about most of these rare tumors. In terms of the TM samples, there are 3 time points. We'll take archival tissue, and a couple points about the vials and how much blood. And really just want to thank, kind of go through this slide and kind of thank everyone. I think the support from SWOG has been phenomenal, especially on the operational front and the statistical support. It's just been a lot of work, the data coordinators, and we've just had a lot of support from the NCI and CTEP as well. In fact, there was a CTEP meeting earlier this week, and this is one of the protocols that was discussed. And so I'm not quite able to tell if anyone from the data team or stats is in the audience, but if they are, I'd like to at least invite them up to take credit for all the hard work they do. And then I'm happy to take any questions. Great, thank you.